Good morning, everyone. I'd like to echo Michael's comments that it's extremely nice to be back in person, and I'm thrilled to do so myself and be here with everyone. It's my pleasure to introduce today Dr. Barbara Stranger. She is an associate professor in the Center for Genetic Medicine and the Department of Pharmacology at Northwestern University's Feinberg School of Medicine. She is an expert in the genetics and genomics of sex differences in human complex traits and diseases. She received her bachelor's degree from the University of Chicago and her PhD in population genetics from the University of Montana and the Max Planck Institute for Chemical Ecology. She completed postdoctoral training at the University of Barcelona and the Wellcome Trust Sanger Institute in Cambridge. She has held faculty positions at Harvard Medical School and the University of Chicago. Dr. Stranger serves as the chair of the Sex Analysis Working Group of the NIH-funded Genotype Tissue Expression Consortium and co-chairs the Sex-Specific Cross-Disorder Working Group of the Psychiatric Genomics Consortium. Today, Dr. Stranger will be presenting on the impact of sex on gene expression, disease, incidence, and mortality. Please join me in welcoming Dr. Barbara Stranger for what I'm sure will be a fascinating presentation. Thank you.
All right, thank you for that kind introduction and the invitation to be here. Likewise, I haven't been in front of a live audience in quite some time. In fact, when I went through TSA, I had a Swiss Army knife in my backpack that I didn't even know I had because the last time I used it was for camping, not work travel. So it's really nice to be here. Thank you. So I'm going to talk to you about sex differences in human health and disease. The talk is going to follow, wait, it's not changing the slide here. Oh, yeah, now it went, now it's not going back either. Sorry, there we go. Okay, let's see how that goes. Sorry about that. So the talk is going to follow the outline that you see here. So I'm first going to talk to you a bit about the introduction, really, about sex differences in human health and disease. I'm going to tell you about the history of sex differences research, some of the hypotheses surrounding the origin of those differences. I'll talk to you about genetics of complex traits and the role of sex there. So it's a little bit of biology and genetics here today too. I'm going to tell you about this big project called the Genotype Tissue Expression Project and give you some ideas about where we see this field going. So first, an introduction. So when people think about possible sex differences in human health, the first thing most people are thinking of is differences in prevalence or incidence of disease. So I think a lot of people know that autoimmune diseases are more prevalent in females, but there are quite a few other characteristics of health and disease that vary between males and females. In disease, age of onset can be different, the way the traits are clinically presenting, so the exact diagnostic criteria could be different for males and females. Disease severity, progression, prognosis can be different. Overall susceptibility, response to treatment, so pharmacology, and even adverse effects from pharmacology. All right, so thinking about sex differences in health, Alzheimer's disease is the most common form of dementia, and 2/3 of individuals with Alzheimer's in the US are female. It ranks as the 5th leading cause of death in women, whereas it's 7th for men. A specific genetic variant in the gene ApoE, this particular genotype, is the strongest known genetic risk factor for late-onset Alzheimer's disease. And the frequency of this genotype is the same between males and females, but the risk of disease in carriers of that variant is 4 times higher for females than men. So it's very deterministic in females, not so much for males. There's no current treatments for Alzheimer's, so this is really a case of societal burden weighted heavily towards female burden. So in the case of depression, it's more common in females, different symptomology, different challenges that come with it. And, it's not all about diseases that, of course, I'm just giving you a couple examples, but not all are about morbidity and mortality. But there are other cases like the autism spectrum disorders. They affect 4 times as many men as women. And the autoimmune diseases typically are more female biased. So you can see just these incidents and some of these characteristics and these traits. And I'm going to walk you through a few others. So first, we're going to look at cancer incidence. So in this panel on the right, you can see the male-female incidence across 13 cancers, ranging from thyroid cancer, which is much more prevalent among females, to the upper gastrointestinal tract cancers, which are much more prevalent in males. In general, almost all cancers are more prevalent in males. However, for a given patient diagnosed with cancer, biological sex can also impact outcome. So in the 2 figures here, I'm showing the sex differences in incidence rate and age-adjusted hazard ratio, or survival, comparing males to females for different cancer types. And so when the incident rate or hazard ratio of survival is larger than 1, so over here, these are the male-biased cancers. And on the other panel on the right, it's the survival. And so, for example, you can see in the very bottom cancer here, this Kaposi sarcoma, this is a lining of the blood vessels. It's much more prevalent in males, but females do much worse. So that's what that bottom row is showing you. You can see in the cases of breast cancer and that's thyroid cancer, it's much more prevalent in females, but females do better, have better outcomes. And so, it's kind of an interaction between the actual disease and the individual's sex. So I said that there's sex differences in response to treatment, and what we're looking at right here is a diagram of a receptor. And so this is the EGFR receptor. It promotes tumor growth and spread in cancer. And so there's therapeutics that have been developed to stop the activity of this particular receptor and therefore stop the cancer. So clinical trials have tested the effectivity of these of the different drugs that have been developed to block that whole chain. And what you can see in this is that the drug is effective. The drug is effective in everyone. That's these numbers you see here, males and females, but it's much more effective in the females. And this is measured by progression-free survival. So that males and females treated with the drug eventually die at the same rates. That's what's shown in the overall survival panel here. But females have a longer period without progression of disease, so longer quality of life. All right, so in terms of sex differences in adverse drug events, women have a 2-fold greater risk for adverse drug effects, and they're more severe. So hospitalization is much higher in females than men, and mortality is, I think, relatively high for adverse drug events. And so this is a real challenge. One of the reasons for this is thought to be that many of these drugs were developed before women were even involved in clinical trials, and so they were never tested in females. And that's not the case anymore, of course, but I'll get to some of that as we go on. And the idea behind why this might be caused is differences in pharmacokinetics, pharmacodynamics, immunology, and also genetics. And I'll talk about some of these ideas a little further. Okay, so moving on to cardiovascular disease. Heart attack mortality differs. For heart attacks in people over the age of 45, 23% of females die within a year compared to 18% of males. And within 5 years after a heart attack, there's a difference in the percentage of people who die, 36% of males versus 47% of females. And females have heart attacks at older ages than males. They're more likely to die of them as well. Stroke is an interesting example because there's an age by sex interaction on mortality. At the oldest age, females are more likely to die following stroke than males. And in some categories, mortality is approximately the same for males and females with stroke. But between the ages of 45 to 74, women have substantially lower risk of stroke mortality than males. This is an example looking at effectiveness of drugs in glioblastoma. So this is a study that was done in mice, and the top two panels are one particular drug, and you can see the two different colored lines comparing the effect in males and females. The two different lines are the control and the drug, and you can see in this case that the drug is effective in males, the longer survival. When you look at a different drug on the bottom, you can very easily see that there's a different drug works better in females. And so this is, these kind of preclinical research are quite important to do and do in both sexes. And this is not something that has typically been done. And I'll talk about that as we go forward too. Now in this next panel right here, these are human tumors that were characterized by the basis of the genes that they have turned on and turned off, kind of like a dimmer switch, so how much of each gene is being expressed. And they were clustered, individual people's tumors, clustered into 5 different groups, the different colored lines you see there. And you can see the survival in males and females. And you can see that it's a different cluster that does better depending on whether you're male or female. So there's different genes are contributing to what that signal is. All right. So sex differences in lifespan. Well, worldwide life expectancy for women is greater than men. I'm sure everyone here knows that. This observation is robust against diets, environment, and demography. So women have longer lifespan, yet more morbidity overall. Why is that? Well, it's thought that that's really due to the fact that men die younger, and so the older remaining men are healthier. And also because the types of health problems with older age differ by sex. So women have more joint and bone problems because of anatomical differences between males and females. In the pie chart, we're looking at the percent distribution of the 10 leading causes of death in males and females in the US. This is telling us that among male or female deaths, the leading causes are relatively similar. This does not tell you about the likelihood of death given a male or female having those diseases, but I think I've shown you a few examples where we can see that. Moving on to, oh, it skipped one. Okay. Well, I think it skipped one, but I was going to tell you about COVID-19. So as everybody knows, we're in the pandemic of COVID, and when you look at the number of people who've been infected by COVID, it's much more male biased already, and but the outcomes are much worse for men too. Approximately 1.7 times higher death rate, case fatality rate in males than in females. So that's pretty stark. And trying to understand why that is, if you think about how the whole COVID infection proceeds, first there's the virus getting into your cells, and then it's the activation of your immune system, first the innate immune system, and then the adaptive immune system and all. And there's lots of pieces of those particular responses. I mean, at every stage, there are things we know about having some sex differences. So I think there are a lot of people studying this right now. In fact, it's just an explosion of science, obviously, around COVID right now. But the sex differences piece is something that we'd like to understand much better. All right, so getting into the history of sex differences research. Originally, women's health was viewed as reproductive health, and just everything else was assumed to be the same. In 1990, the Office of Research on Women's Health was established at the National Institutes of Health. in response really to congressional, scientific, and advocacy concerns that a lack of consistent and systematic inclusion of women in NIH-supported research would lead to clinical decisions being made about healthcare based solely on findings from men without any evidence that they were applicable to women. So that was a big step. In 1993, a federal law was passed to mandate inclusion of women in clinical research. Before that, there were reasons about trying to protect females. They might be of reproductive age. They didn't want them in clinical trials, pregnancy, all that. But it's really important that everyone is represented for I hope, obvious reasons. But it wasn't just in clinical trials. In the preclinical work, as I showed you before about mice work, for example, the field has very, very heavily only worked with male mice. And in the plot you can see here, this is a review article that was done in 2011, looking at the inclusion of male and female mice across different disciplines. And you can see that in some of them it's very, very stark, the percentage of male animals used. And this is 2009, that's not that long ago. And in fact, it still persists today. And the reasoning behind that is that investigators think that having females involved makes too much variation. There's hormonal cycles, and they just think it's too complicated. And that they can have a larger, more homogeneous pool by just doing males in their studies. So because of that, the NIH passed a new rule in 2016 specifying that any government-funded scientific research has to consider sex as a biological variable. And it doesn't mean that everyone has to do sex differences research, but it means that at all levels of clinical research, preclinical, clinical trials, everything, there have to be both sexes involved, unless it's a sex-specific trait, of course, and that you have to do that, and you have to justify it, and you have to make the data available in a way that it could be disaggregated or studied separately. So this review article also had a study showing study showing in these different fields were the data, even if they had both sexes, were they analyzed by sex? And you can see that it varies a bit by traits and so on, or not traits, but by disciplines. And certainly some of them never even look for sex differences. I would say that is the vast majority. I mean, and certainly in the research that I work on. So it's really important. So this sex as a biological variable, or SABV, can really yield new knowledge. So this is an example about stroke. So there's higher incidence of stroke in men, but the outcomes are better in men. And what happens in stroke is there's ischemia, loss of oxygen, and then it produces nitric oxide, and then there's this molecule, PARP1, that starts promoting cell death, okay? Which you don't want. And so this whole pathway was identified using only male mice. Now the investigators with the data on the right wanted to see how well this holds in females and males. So they went and they applied a inhibitor of PARP, and they wanted to see the outcome. And so if they aggregated the individuals together, it would say that this molecule didn't do anything. But what you can see if you disaggregate the data is that it's extremely effective in males and not in females. So that led to a whole different investigation of what is the pathway for this in females. And the thing about finding a sex-specific pathway is it means then you can come up with a different therapeutic, right? And it can be part of personalized medicine. These ideas of finding something that suits you best, your person. All right. So an example in pharmacology, a lot of people know something about Ambien. And what happened is after Ambien was approved, the FDA learned that women were at higher risk for excessive daytime sedation and driving impairment. Researchers investigated the underlying causes and were able to demonstrate that females have higher drug exposure than males after taking the drug, and it couldn't be explained by body weight differences. So they confirmed slower elimination of the drug is what was leading to the extended time of the elevated blood levels of this drug in women. And then they announced a 50% dose reduction for females. So had this sex as a biological variable been incorporated into the studies of Ambien, this observation could have been noted and these deleterious effects avoided rather than post hoc discovery. In the review of literature, for most FDA-approved drugs, elevated concentration and longer elimination times are strongly linked to adverse drug response in women. In general, though, there's pretty terrible reporting on this, and most FDA drugs were approved, as I said, before women were required to be in clinical trials. All right, a couple other examples from pharmacology. So there's a whole series of enzymes called the cytochrome P450 enzymes, and there's, as you see on the table on the left, there's a set of them, different ones. And the activity of these enzymes differs between males and females for several of them. And then not only the activity of them, but also on the right, what you're seeing is males and females produce the enzymes at different levels. And so you see some of them highlighted in pink and blue, and it's sort of which is higher in males, which is lower in females. And these three that you see highlighted in the box, these are responsible for metabolizing hundreds of FDA-approved drugs, so higher activity in female and male, so those pink ones anyway. So, you know, it matters. Okay, cardiovascular disease. Younger onset in males than in females. And, did I skip one? Huh, okay. Well, sorry about that. I think something got skipped. So just back to the clinical trials thing. The issue about clinical trials, even today, even though these requirements are there, they're diseases that are disproportionately biased towards females, and yet we're still underrepresented in the clinical trials. So these are new data. This is the picture from today. Got it. Here's the cardiovascular disease that I was trying to say. They have younger onset in males, older onset in females, and the type of cardiovascular disease is different. So we see more obstructive coronary artery disease in males and more microvascular dysfunction in females, and the death rates are different as well. So trying to understand this, people investigated studies of the heart turned out to show that there's differences in the architecture of the vessels. And so there's differences in diameter, different branching patterns, and so that leads to different patterns of disease and blockage. All right, so let's talk a minute about what are some of the hypotheses surrounding the origins of sex differences. Now, this is a really busy slide right here, and so I don't want to go through all of it really, but it's thought to be an interaction between genetics and the environment. So the genetics piece starting out with is we have sex-determining chromosomes. Females have 2 X's, males have an X and a Y. And so that right there starts everything from the beginning of having different genes turned on at different stages of development, and it has all these downstream effects. That's just the starting point though, but then there are more differences, of course, in our gendered environments, which is different than biological sex, but exposures we have could be through occupations, it could be through things we ingest, as shown in this slide here, but there's also the endogenous environment where we have different hormones and different levels of hormones and so on. So how that can all manifest is it turns into essentially our genes being turned on and turned off and amplifying these sort of signaling cascades of genes being turned on and off that are very different between males and females in some cases that I'm going to describe to you a little bit as we go forward. So I'm going to pivot a minute and tell you guys a little bit about genetics. So some fun facts about the human genome. I don't know if you guys will think they're so fun, but I hope so. So the human genome is 3 billion base pairs of DNA. And so in the picture you see on the right, the base pair, it's arranged in a double helix. The DNA is then compacted, like tightly wound into the chromosomes. We have 23 pairs. You can see at the bottom the XY and the XX, those are the sex chromosomes. There are approximately 30,000 genes, and each gene is like a recipe to make a protein via intermediary messenger RNA or mRNA. That 35,000 genes only comprises about 5% of the genome. The rest of our DNA is not genes. And that was a pretty surprising finding at the time, because then what people said is, what is all the rest of that doing? And we're still trying to understand that, frankly. A lot of it is how we regulate the genes, turn them on and turn off. There's quite a bit of machinery to do that. So this at the bottom here, this is kind of the central dogma of genetics, is that DNA gets transcribed into mRNA. We refer to that as transcription or just gene expression, and then that becomes protein. And it's kind of like a dimmer switch. It's sort of how much on, how much off. All right. So human genetic diversity. We're actually not all that diverse a species. We're 99% genetically identical to each other. And when I say genetic diversity, I'm talking about the DNA sequence being different between each other. So even though we're 99% similar, there are genetic variants, and so there's multiple variants of any gene in a human population. Many genetic variants are shared across individuals, but we also, each one of us has millions, possibly, of unique variants. And this genetic variation can impact all of the different traits: hair, eye color, height, disease risk, all kinds of things. So back to that central dogma idea, genetic variation can impact trait variation, as I just said, but how does it do it? It can be through changing the sequence of a gene, of course. It could change the sequence of an actual gene, then you're making a different protein. But much of it occurs through effects on gene regulation. And so genetic variation can impact levels of a gene expressed, ultimately impacting the levels of protein. And when we talk about this, we talk about then the messenger RNA or the protein as kind of an intermediate trait between an individual's genetic sequence and their propensity for, in this example, a given disease. So, but I mean, even though this example is about disease risk, it could be a drug metabolizing trait like we were talking about before. Someone could be a fast or slow metabolizer based on how much of that enzyme they make. All right. So in complex trait genetics, we're trying to understand the genetic basis of traits that come from an interaction between our DNA sequence and the environment. We link genetic variation to the trait, and in doing so, we ask a number of questions that you see here. So where in the genome is the genetic variation that impacts this trait? Is it in a gene? Is it outside of a gene? It turns out that most of the genetic variation influencing disease is outside of genes. We can ask whether a trait is due to only a few genes or many genes and different effect sizes. And it's challenging, though, because even if we get to the spot where we think we know there's a genetic signal in the genome for a disease, then, as I said, most of that's not in genic regions. So then we say, well, what's it do? And then we have to do a lot more work trying to understand what that particular genetic mutation could be doing. So why do we want to do this? Well, besides just the fun of understanding how our genome works, which for me is a lot of fun, there's some real practical things here. And so one thing is for drug development. If you can identify genes or biological pathways, that's a means to identify putative drug targets. And given the rate at which clinical trials for drugs fail, it's turned out that gene-based targets identified through genetics, are twice as likely to make it through clinical trials. This example that I put here, PCSK9, is kind of an interesting one because this is one that is involved in LDL metabolism, so that's the bad cholesterol. And so researchers essentially found experiments of nature, basically people and families that had mutations that either lowered the cholesterol or made it very high. And so the people who had lower cholesterol, they had it lifelong. And so they found more people that had that. And they saw that people had really reduced risk for all kinds of cardiac events. And so drugs then were developed to mimic that, like anti-PCSK9 drugs. And so those are FDA approved now. The other thing is predicting risk. Just as there's always been algorithms to predict risk of heart attack, like based on age or sex or your family history, genetic risk can now be part of that puzzle. And so by understanding the genetic basis of a disease, your DNA could be read and you could be assigned a risk score. And the thing about that is, why is that valuable? Well, it depends. If there's something clinically actionable you could do about it, in this case, if you knew you were at extremely high risk for high BMI, you could modify your environment, diet and lifestyle choices and things. But if it was for a disease that you couldn't modify your environment, maybe you could modify being screened for it and catch it earlier than later. So this is not quite implemented yet, but is certainly way of the future. All right. So in genetics, certainly males and females have been analyzed together as a single group. And historically, the sex chromosomes were excluded. Part of that just has to do with it requires different statistical models to analyze them. It's very technical, so I won't get into that. But they weren't developed for a long time, and everybody just left it out because it was a nuisance. And generally, that's the whole thing about biological sex. People sort of think of it as a variable to just even everything out and get rid of that effect, as opposed to trying to understand it. So as people in the last 10 years or so have appreciated that the effect that a specific genetic mutation can vary with context. So like a resting cell versus an activated immune cell, a genetic mutation might do something different in those contexts. So as people have recognized that there's context specificity of those things, people have started thinking about sex as a context that interacts with sex. Okay, so I showed you this slide before. What is the origin of the sex differences? And so I'm going to tell you about a study where we asked these questions. How different are males and females at the level of gene expression? And do the genetic variants have the same effect regardless of an individual's sex? So when I say same effect on gene expression. And I'll tell you about the NIH GTEx project right now. So there we go. So this is a project called the Genotype Tissue Expression Project, and it was launched in 2010. The goal was to do a standardized collecting of 50 different tissues from 1,000 different individuals. So these are post-mortem donors, and they all had their genome sequenced, and then gene expression of all those 35,000 genes measured in each one of those 50 tissues. So the primary scientific goal was to understand how genetic variation can impact gene expression variation. In that little cartoon slide there, you can see that this genetic variant, so two different versions of it, turn out to produce different levels of the mRNA for that particular gene. And so by making those maps, you can integrate that with the mapping of disease. So you could try to make these links between a genetic variant that is associated with disease and impacts a particular gene. expression level. And so that kind of makes this hypothesis that the disease is caused by variable expression of this gene. So that's the kind of thing that we're doing with this. And this is way beyond the scope of anyone's individual lab could ever do this. It cost a lot of money. But the other piece of this is making these publicly available resources for investigators to use across all kinds of disciplines. So my lab worked in this project, and we led the group that looked for biological sex differences in these tissues. And so the first question was about just differential expression. Do males and females have different expression in these tissues, and how much, and that kind of thing? In the end, that project didn't have all 1,000 individuals. It had 838. But we had a lot of tissues, and we tested all the different genes. So what did we find? So we found that in any given tissue, which are the different bars you see on the left, don't worry about the abbreviations of the names of the tissues on there. I'm sure they're a little small to read anyway. But so what we found was that in a given tissue, somewhere between 475 about and 4,500 genes per tissue were differentially expressed, so different levels. That's 1 to almost 13% of the genes. Overall, across all those tissues, 37% of our genes were differentially expressed. Most of that was not coming from the sex chromosomes, I mean the vast majority. And one thing to note is that we were surprised, really, by this. We really didn't expect that many genes to show that kind of bias. But the differences were small, and it's a little hard for us, just with that kind of information, to understand, like, does it matter? What are these doing? All right, so we asked the question about, okay, so is it the same? I mean, the numbers were different across the different tissues, but are they actually the same genes? And so we saw that 18% of the differentially expressed genes were expressed in only a single tissue. That's that top bar you see up there. That's the tissue that was, or genes that were expressed only in a single tissue. And you can see how that distribution goes to 44 at the very bottom. So there were very few that were differentially expressed across all 44 sources, and those were from the sex chromosomes primarily. So this was also interesting. So that means that the differential expressions that's happening is happening in a very tissue-specific way. So individual tissues could be very biased to some set of genes, and then another tissue could be biased to other genes. It's not that surprising. I mean, gene expression does vary by our tissues. That's why they have different functions and all that. But this particular program of sex differences is quite interesting, and we didn't know anything about this before. So we can ask this question, okay, so there's a lot of genes, a lot of sex bias, it's different in different tissues. What kinds of functions are we talking about? What do they do? And so we can do this thing called gene set enrichment analysis, where you take a list of genes that are, and list them, rank them by bias and the effect size. And then you can look at a database of known gene ontologies. So these are annotating genes for what's their function. We can do a statistical analysis to ask, are differentially expressed genes enriched in any of these functions? So that's what we did, and this is what we found. So we found that sex-biased genes are involved in many important functions and overrepresented. So that means they're representing specific functions more than you'd expect by chance. And so we have enrichment in genes involved in drug and hormone response, embryonic development, tissue genesis, fertilization, a whole bunch. So cancer, fat metabolism, immune function. So really important functions, and that are really expressed differently in males and females. So, and there's disease genes in here and everything. So quite interesting, not just random. All right, so just to wrap this piece up, we saw a lot of sex differences in gene expression across human tissues. The tissues differ qualitatively and quantitatively, so which genes they are, but also how biased they are. We saw these really important functions of these genes, and also, disease genes. That's something worth looking into even further. Okay, so we also took a look at, the genetic effects on gene expression levels. So I showed you that little cartoon that's on the left already. And so how do we do this? We have a group of individuals. We get all of their genetic variation information. We get expression of all their genes. And then we do a statistical association. So in that plot on the right, we're just considering a single gene and a single place in the genome. And a person could have a genotype of CC, CT, or TT. In this case, what we're seeing, each dot is a person. We're seeing that people with the TT genotype have higher expression than people with the CC. So the T is conferring higher expression. And so when we do this kind of a study for one gene, we walk through the genome, like at variant by variant by variant, doing a test like this. So we do millions of tests, up to maybe 10 million tests per gene. And then we do it for the next gene and the next gene. So 35,000 times 10 million. So that was what the GTEx project was doing. But we wanted to ask the question of, okay, so if we find those kinds of associations, is it happening the same way in males and females? So could we find genetic regulation that acts in a sex-dependent manner? So if you look at these cartoons here on the bottom, we're looking for cases like these where the effect that I just showed you in the previous slide is present in only one sex, or it's an opposite direction effect across sexes, or it's present in both sexes, but the magnitude of the effect is different. So on the right, you see that different slope between those two. So we did this across the GTEx tissues. I'm just going to just show you a couple examples here. So in the panel on the left, you can, well, actually, let me just orient you to these three panels. So each group of three has the males on the left, the females in the middle, and a combined analysis of everyone together on the right in gray. And so this very left panel of three, you can see an association that's present in both males and females with the different effect size in females, bigger. And if we analyzed everybody together, we would still see that there is that association between that particular variant and the expression of that gene. But what we wouldn't know is that it's a different effect size between males and females. Now, the next panel you see is opposite direction effects. So this is the same genetic variant that one of the alleles is expressing or makes the gene expressed higher in males, and that same one is expressing lower in females. Exactly the opposite effect. And when we put everybody together, it looks like there's no genetic regulation. So this is the kind of thing we don't find if we just do analysis of males and females together. And the one on the right, this panel, there's no signal in the males. There is a signature in the females. And there's a signature in everyone together, but what you wouldn't realize there is that it's driven by one sex only, that it's really a sex-specific signature. So we did this across all the tissues of the GTEx program, and this is what we found. We found 366 total genes that had sex-biased genetic regulation. You can see that it's very uneven, what we're looking at here. The big turquoise line here in this graph is breast. So breast tissue had the most, but there are other tissues. Skeletal muscle is the next one. Now, it seems like there's not much here, but we really think we're just scratching the surface of this because it's a very underpowered statistical analysis we're doing. It's an interaction analysis where we're doing essentially an individual's genotype by sex interaction, and we're doing millions of tests, as I described earlier, so we're very underpowered. But the fact that we see this is quite interesting. And so if you remember, I talked about considering mRNA as an intermediate trait between possibly disease and genetics. So we tried to assess that, and we had information, genetic mapping information for 87 traits. Many of them were diseases, and we have these genes that have this differential by sex regulation, genetic regulation. And so we applied a statistical model to test whether the same genetic variant impacts the expression in this differential way and impacts disease. And so what we found was 74 new associations between specific genes and specific traits, and 58 of those were sex-specific. So that's really, it's a great discovery. I mean, given we only had that many genes even to start with, this 366. But so this is really new, brand new biology, puts new genes forward towards being causal genes for these traits. Now, we also saw gene-trait associations that had been previously detected using combined male and female samples, but now what we can see is that actually that association is only valid in a single sex. So it's a little bit like some of the examples I showed you a bit earlier. So considering sex as a biological variable leads to both new associations and greater resolution of previously identified associations and discoveries. So just to summarize this piece, we do see that there's evidence for sex-biased genetic regulation of gene expression. I mean, this means the exact same variant is doing something different in males or females, and it's quite interesting. We've been able to demonstrate that some of the previously identified examples of genetic regulation were really due to signal in only a single sex, and as I said, we can discover new links between disease genetics, genes, and mechanisms being the expression of the gene, regulating the gene. And so looking forward, you know, I hope I've convinced you all that biological sex is really a fundamental feature of human biology and human health, disease, and even mortality. And there's a lot for us to understand, to try to really get at what's causing this. And so we went really to this very fine molecular scale of what's happening in our tissues and can see that there's quite a few sex differences and across every single tissue we've looked at. And so there's quite a lot for us to understand. We can also see that genetic variation interacts with biological sex, and I've shown you, and there's got to be so many more examples of how biological sex, or cases where we really do need to disaggregate data, include males and females, and really look for these differences, because we could really be missing a lot. And it's actually quite, I mean, if you have the data, it's really easy to do that. Why not ask the question? In every study, pretty much, of human biology, if you've got males and females, it's just one extra analysis to do, and take a look and find out. So I do think that research like this is going to end up helping us with this goal of precision or personalized medicine in the future. And I'm excited to be a part of that. So with that, I will just thank some of my collaborators in the consortium, as well as lab mates at Northwestern, and of course the donors who donated samples, and their families, because these are post-mortem donors. It's a big commitment of someone to do that. And then finally, thank you all for your attention. Thanks.
We'll wait just a second to see if there are any questions. And if anyone has questions today, we're not passing the mic around, so you need to just go to one of these stationary mics and ask any questions that you have.
Thank you very much. Really interesting. I just have one question. Do you think that, I mean, it seems like there's a lot of more research taking place, and this is kind of an early stage. Is it because you don't have enough data? I mean, you had like one study with 1,000 individuals and so on, or is it like, so it's going to take a lot more time for you to have more individuals?
Well, so there's a few different things here. The first, I would say, is there's a lot more research to do, because it's really brand new that people are appreciating the value of this kind of research, right? I mean, that is very new in and of itself, to even bother to look if there's sex differences in almost anything we study. And in fact, you know, when I started doing this work, I used other people's data sets because I was a new investigator. I didn't have that much funding for my lab, and so I went to some of my senior collaborators and said, hey, can I look at this in your work? And everybody pretty much said yes, because they said, we don't care. Yeah, go ahead. And so I think that there's that piece of it. And now the thing that I said about the discoveries, that I said underpowered, yeah, we really do need bigger sample sizes. And this study that I just presented is the biggest study of that kind anywhere in the whole world. Like, this is the first data set that was collected across a single cohort of people where, you know, we could really measure these things at the molecular level, but from the same group of people. Before, people had said, oh, you know, we have this sample of skin from this group of people, and we have this sample of skin from this other group of people, and we would compare something between them. But you wouldn't, that would be a difference between sort of two populations. But this way we could look across tissues in the exact same set of people, and it just doesn't exist. It costs a lot of money to do this kind of research, and everything's increasing now, and we can meta-analyze with other places and so on. I mean, there's a lot of investment in big biobanks now across the world, huge biobanks, and so we can start doing meta-analysis across those, and we'll have better power to look at these things in the future. But really, it's an environment for low-hanging fruit in a way right now, because people just haven't looked for the most part.
We have a question coming in online. For people who self-identify as non-binary, will those be excluded from this type of research, or will they be allowed to pick which way to classify or what?
Yeah, you know, I should have said something about that. When I refer to biological sex, we're referring to the sex chromosomes, and usually biological sex is sort of sex assigned at birth. And it's not based on chromosomes, it's based on anatomy. In our studies, because it's genetics, we were doing XX versus XY. Now, we don't know anything about how people self-identified in this particular study. And we also don't have anybody who has a different chromosome composition of X and Y, because there are. It's not actually that small a number of people who have maybe an extra X or extra. And some of these have associated disorders, and some don't. And so this study was limited that way. But the study of gender, I think, is something different, and because that's really a social construct. And there's research about gender differences too, of course, but that's not what ours was. And so I think if we're doing a study of gender differences, absolutely, your self-reported gender is what matters. Anybody else? All right, thank you very much.